I love these critters. All right, weird critters. Anyway, like I was saying with that video that I showed earlier, the flagella was on the back of the protozoan. The protozoan was moving in such a way, I should have showed you that. But remember, there are some protozoans that the flagella is on the front. For example, in the Euglena, they actually have two flagella, a bigger one and then a littler one. But those are on the front of the organism. That's going to become significant. So that flagella, if I could make this statement, this is just going to be kind of one of those like, what? But that flagella that we were looking at under the microscope there just a few minutes ago, because its flagella, or that protozoan, because its flagella was on the back, I know that relatively speaking, among things with flagella, that thing is more closely related to us than things to have their flagella on the front. That a weird statement? So looking at that thing under the microscope that Alexis shot in our drop of pond water, that is more closely related to you and I than, say, the Euglena, whose flagella was on the front. Because if you think about it, there's a cell out there that we have, us animals have, in which the flagella is on the back. There's only one, flagellum, excuse me, flagellum, oh man, flagella is plural, flagellum singular. What is the cell that us animals create that only has one flagellum? Yeah, see? See, look at that. See that connection? See, so what that means is you got a similarity there, right? And we've talked about how similarity, now sometimes similarities can fool you, right? Sometimes similarities can be because of convergent evolution, but in this particular case, it's not. So that starts off with a very interesting sort of idea that we're dealing with here, that there is a protozoan that we are more closely related to than we are to other protozoans, and that kind of highlights what protozoans were. Now, the first thing that we thought protozoans were was small, and that's not a very good way to classify things, is it, right? As a matter of fact, and it's gone now, I don't think anybody uses it, but years ago when I was learning about the kingdoms of life, for one thing, we didn't have domains back then. We just had 5 kingdoms. All the prokaryotes were stuck in one kingdom called Kingdom Monera, which is way wrong. But the same thing with the protozoans. We lump them all together in the kingdom Protista. And that kingdom is only slowly beginning, well, not slowly, has only recently begun to die off. In other words, people are saying it's no such thing as the kingdom Protista. I'll show you in the next slide, I'll say it again, but the kingdom Protista is polyphyletic. So why did we get this idea that they were all in the same kingdom? Well, because we were doing what we always have done as humans. When we were looking under a microscope, we said, what the heck are these things, right? Robert Hooke, that dude up in the upper right, is one of the first folks we credit with looking under the microscope. He was looking at cork cells. There's a drawing. It's where we get the name cells, right? Right? He said, oh, they look very regular. Thank goodness he was looking at plant cells. If he'd have been looking at animal cells, who knows what he would have called these things? Because animal cells don't have those regular shapes, but the cork cells did. So he said cells. That's a good word for them. All right. And about this time, a contemporary, his 1635 to 1703, Robert Hooke, 1632, 1723, von Leeuwenhoek down there in the lower left. Little nicer looking fellow, apparently. But there's his microscope. I love how these old things, they made them so super elaborate, like much more than they had to be. Like they really went into the artistry of the instrument they were making. But what I'm trying to get at is, if you have to think about that, some of our discoveries in science are very closely related to advances in technology. So in other words, we really didn't know what there was in the old drop of pond water until we could make microscopes that were good enough, made well enough to see that. Like we couldn't really know about, there were other, up until the 50s, we didn't know there were other galaxies out there. We thought that the Milky Way was the only galaxy. And then we started making Bell telescopes, and we went, whoa, holy cow, there's a whole bunch of galaxies out there, right? Same thing with looking at stuff in a drop of pond water. That's the whole thing behind, understanding where disease came from when we were talking about prokaryotes. Couldn't see the doggone things. And those, remember, prokaryotes are really small. When von Leeuwenhoek looked under a microscope, he didn't really know what to call these things. He didn't know if they were animals, whether they were plants. So what do you do when something you got doesn't fit a category? Make up another category, right? Maybe it's a new category. So he called them animalcules. Animalcules is animal-like things, but they weren't really animals, at least as far as we knew. But he didn't know what they were, so they called them. I love it, a little quote there. People always say, "What's one of your favorite quotes?" You know, like in in general, and some of them are things I can't repeat because they're funny. Anyway, funny little limericks. But this, I love this quote. "No more pleasant sight has met my eye than this," von Leeuwenhoek writes, when looking through the microscope for the first time and seeing protozoans. I just think, wow. Now, that's so reserved, right? Von Leeuwenhoek, so he was probably German, right, with that name, von Leeuwenhoek. So, rather European and very reserved, and, he's dressed like he probably was, if you studied science back then, you weren't eking out a living, right? You were doing decently because other folks were living on subsistence farming. So, von Leeuwenhoek there, I can imagine him looking through his microscope in very proper German, right, making this statement. And to me, what I've always loved about the statement is it captures that moment when you go, wow. And it doesn't have to be looking through a microscope, and it doesn't have to be looking through a telescope. It can be taking, if I could stretch it. It could be in the middle of an accounting class. It really could. You could say, wow, I love this. Look at this debit and ledger stuff. It just gets me, right? But what I'm trying to say is, whatever does that to you, that's where you need to go in life. Whatever makes you go what von Leeuwenhoek did that day when he went, whoa, look at this. Or like when I was a little kid and my dad took me to go see this movie about sharks, and I just went, wow. Fish, and it just hit me like that. So it may have, yeah, there you go. Sean's got it exactly. So it may have already hit you, right? And if that's the thing, you follow that. You make that thing. You follow that thing that makes you say what von Leeuwenhoek wanted to say. And folks, I'm telling you right now, if I get in a microscope, put down a drop of pond water, I still can get that feeling. I looked, and you gotta find that job that does it. Now maybe you weren't quite as jazzed up as I was the other day underneath looking under the microscope, and that's fine. We're all different things, but that's what you got to do in life. You got to find what makes you say that kind of stuff, like, whoa, God, this is so cool, and makes you say it the rest of your life. My son asked me one time years ago, how do you go through life with a job and don't get bored? And I said, you find one that doesn't bore you. And I know that seems silly, but I don't know. I just haven't been bored by this ever. I've been teaching this stuff for 100 years. All right, let's look at these weird. So, Old Kingdom Protista polyphyletic. Y'all remember what polyphyletic means? Someone tell me. What does polyphyletic mean if we're talking about cladograms? Or what does it mean about, right? So polyphyletic is a term that is very similar to flying things, right? Remember when we were looking at, we said it was flying vertebrates. All right, so we'll give you that. But we had a bat and a hawk, right? Flying vertebrates. All right, because they're flying vertebrates or flying things, for goodness sakes, does that mean they are relatively closely related? And I don't need to explain that term now. You understand what I mean? But does that mean because they both fly that they're relatively closely related? Because we've said this is the flying vertebrate group. Versus what if I said the mammal group? No, yeah, right, exactly. Remember, it's all about convergent evolution. So a polyphyletic group is a group that really just is something very artificial, right? We could say organisms that live in the water and call that a group. The only thing about it is, you would not do that because you quite clearly understand that there are fish in the water and there are things there. But if we don't understand enough about protozoans, you could see how initially we were tempted to kind of lump them all together and said they are the small things that live in water. The other day, Caitlin, right, the flatworm, not even a protozoan. Not a protozoan, a complete animal, and not a group we haven't even talked about yet. So calling things small, in a kingdom is very, very polyphyletic. You guys were looking at algae yesterday. Some of it was prokaryotic, or the other day. Some of it was prokaryotic. Some of it was eukaryotic. Some of it was plant. Some of it was protozoan. This is a really mixed up group, okay? So we're going to take a look at each one of these groups. You're looking on the right-hand side, the ones in the brown and the green, these guys right here. Yes, of course not. Work. Thank you. Let's see. There we go. The excavata, the SAR clade. You remember what a clade is? A clade is a group. Doesn't mean it's monophyletic. So you'll notice when you look down here, even now, these little dashed lines. What do these little dashed lines tell you versus the solid lines when you look at a cladogram? What does that tell you? You got the right idea, Sean. It's not necessarily the distance in the relationship, it's actually the relationship itself. In other words, the split that's drawn there may not happen exactly like that. All right. The other ones that are in solid, we have more confidence that that's the way the break occurred. But in this case, like the stramenopiles, the alveolates, and the rhizarians, those all make up the SAR clade, S-A-R. Whether they should belong together, in other words, whether they should all come off of one common ancestor, is not quite certain yet. There you go. Could be a big uncertainty, right? Right now, we're sticking them in the SAR clade, and it could be, we could find out that sticking all these critters that we're going to go through into the SAR clade is like saying flying vertebrates. You know, at a different level, I understand, because these are protozoans, but remember, the more you know about them, the more you realize, man, these things are really, really different. Like we did with the prokaryotes, remember? We separated them into two domains. They were so different. They're so different that there's more differences between the two domains of prokaryotes if you think about the way we use cladograms, right? Than there is between you and the kingdom Plantae, because y'all, we and them, plants, us, we're all in the same domain, right? That means that, relatively speaking, we're more closely related. All right, let me erase that thing. So we got the SAR clade. We'll go through the archaeoblastids, which is the algae. Now, look in the archaeoblastids. Here's where it gets funky. Here's where it gets polyphyletic. Among the protozoan group, known as the archaeoblastids, there is a group that gave lineage to the land plants. Plants are not protozoans, folks. Plants are not protozoans. Protozoans are not plants. But there are some archaeoblastids from which we know land plants evolved from that are still, to this day, protozoans. The red algae. Oh, it gets real funky in here. The green algae. Oh my gosh, Aristotle. Let's talk back to him again. Aristotle, here's where your ladder really falls apart. Because right there in the green algae, the chlorophytes and the charophytes, we really get this idea that there's something in between. Because these critters, we'll talk more about those when we get into plants and stuff. These critters, these green algae, they can be everywhere from very protozoan-like green algae to very land plant-like green algae. Remember I said that word algae is very misleading. You'll notice there are, look up here, there are golden algae and there are brown algae, and those are protozoans. And then there's, remember Anabina? With the prokaryote lot, the little things that look like a string of blue-green pearls. Anabina is a blue-green algae, so don't get fooled by that. So you see right away, among a protozoan group, there is a land plant. All right, now we move on down to the another group, the Uniconta. The Uniconta, uni means what? I think I just said it. I didn't mean to say it. What's that mean? It means one, doesn't it? Cont is pole. Uniconts have one flagella. Ah, look where we are. We're a unicont, aren't we? See down there, the animals in the red? Look who else is a unicont, a fungus. Okay, well, fungi are in the kingdom fungi. Animals are in the kingdom Animalia. Plants are in the kingdom Plantae. We all know that. We keep that the way it is. But look in between fungi and animals. There's a protozoan. That's called a quanoflagellate. Now, I'll show you a quanoflagellate either in this one or when we get into the animals. I don't remember where exactly I put it. But a quanoflagellate is a protozoan. Get it? It's a unicont protozoan. But if you look in the relationship right here, it's more closely related to us, a protozoan, than a fungus is, or a land plant. And among the fungi, the nuclearids, these guys here, they're more closely related to fungi than fungi are to animals or to plants. And nuclearids are protozoan. See? The opisthokonts down here, look, we got a little more uncertainty. Not sure exactly when or where or how this happened. But the opisthokonts have their one flagella on the back. All of these we're going to look, the amoebozoans, the choanoflagellates, all have their all have their flagella on the back, just like our sperm cell does. Very distant evidence, but suggestion of similarity relationship. Now you know why there's so many questions about the protozoan jungle. There's many things. Some are unicellular, colonial. Some can reproduce sexually or asexually. They're all over the place. Good ones, bad ones, mobile ones, sessile ones. Sessile means not moving. But more importantly, I tell my students, think of protozoans as being the stem group for all the other critters. So we go from prokaryotes to protozoans, and that's where the diversity comes. Look at all these protozoans, and then off of those come the plants, the animals, and the fungus. So that slide or that chapter I did a few chapters ago had a really nice picture of a bush, and there was a and there's a protozoan down there at the base of it. So it wouldn't be the roots, that'd be the prokaryote, but it'd be the trunk, and the branches would branch out of that. Neat group, man. Neat group, right? All right, now let's look at some of the origins of these protozoans, or some of the way they came about. And it goes back to what I talked about in the semester. You remember there was a time on Earth during 2.2 billion years ago in which the oxygen levels began to rise on Earth very quickly. We called it the oxygen revolution. We give thanks to the cyanobacteria like Anabaena because that's what they did. That was their contribution. They photosynthesized. When organisms photosynthesized, they didn't make oxygen in order to, with the benefit of the animals. That was just a byproduct of the chemistry. Heterotrophs, or animals, just came along and used that oxygen because it's very efficient to do it that way, right? Aerobically than it is anaerobically. All right, so let's take a look at what some of these organisms, how they evolved, and some of the changes that occurred. I mentioned this to you briefly. Well, not briefly. We did talk about this back when we were talking about what happened to some of the prokaryotes. They underwent endosymbiosis. So among the protozoan group, we get these things, these dinoflagellates, these stramenopiles, these euglenids. If you're thinking, where is he saying these words? Over here on the right. The chlorarachnophytes. All of these protozoan groups came about, we believe, through either primary or secondary symbiosis. One more time, endosymbiosis. There you go, Steve. Either primary, like for example, some sort of heterotrophic eukaryote would engulf a cyanobacterium. Now remember, endosymbiosis would only work if there was a symbiotic relationship, there was a benefit. Of course, if a cyanobacterium you have engulfed, you now can photosynthesize. And so then the red algae, they engulfed this thing that actually helped them photosynthesize just a slightly, I don't want to give it away. Red algae did another thing of secondary endosymbiosis. It gave them a special trait. I want to talk about that when I get to red algae. I'm going to do it right now because I don't want to spoil it. And then on the bottom, the green algae, they underwent a secondary endosymbiosis where the cyanobacteria that was engulfed got engulfed again. And we see basically organisms that are engaging in this endosymbiotic relationship that's giving them properties. And this was very commonly occurring among the protozoans. See, this was not a prokaryote over here. This is a heterotrophic eukaryote here at the beginning. And it's engulfing things like prokaryotes, but it can engulf other eukaryotes. So endosymbiosis, and it might even happen more times than that. One of the really cool evidences for that is this thing called a nucleomorph. If you look inside of it, you can actually see that it was a cell engulfed inside of another cell. And you remember the mitochondria from Bio 1? It's double membrane, isn't it? That indicates that it was a membrane engulfed in a membrane, which gives us ideas that it might have been a free living organism. Now, other parts of the eukaryotic cell have membranes in them too, but maybe that's where the double membrane came from in the mitochondria and in the chloroplast. All right, so we're ready to jump in this group, right? Let's look at the excavata group first. Let's go back to the top. So we're in this group that all together collectively we believe fit into the protozoan group, right? Excavatas are made up of three basic groups. The diplomonads. Those words are so cool. If you just, like I said, if you just, go with the phonics of sounding them out. So diplomonads, parabasalids, euglenozoans, all right? All of those fit in the excavata group. Now, they're called the excavata group because they have a groove on one side, like you see in Giardia there up above. All right, they have this groove on one side of, I guess, a body is a really stretch of the term, but they use that for feeding. So we believe they use that for excavating out of whatever it is they're feeding upon. Giardia intestinalis gets its name because it is a parasitic organism, and it affects you in your intestines, and it can actually give you gastric distress. Let's just say, right? It causes stomach problems, diarrhea, that sort of stuff. These diplomonads, like Giardia, can live in crystal clear places. Like in this mountain stream, you would never think that this parasite Giardia, there it is in the upper right, would live there, but they do. They do quite well. That's when they say when you're out hiking that you should always sterilize your water no matter how clean it looks. Diplomonads, DI, that's a clue to something about these organisms that you can recognize them. Diplomonads have two nuclei. See them? Like two little eyes looking back at each other. Multiple flagella. So multiple flagella. Notice where the flagella are. They're on the front. They're not in the back. Right? These aren't the opisthokonts. That's us and the fungus and all those guys. A degenerative mitochondria. Something called a mitosome. So let's do a little detective work. If your mitochondria is degenerative, you probably rely more on this kind of respiration, don't you? Because that's what the mitochondria does. Remember from Bio 1, it allows you to oxidize. Well, if you don't have oxygen, then you do it anaerobic. And if you have a degenerative mitochondria or a mitosome, then you're going to engage in anaerobic respiration. Now, being that these organisms are parasitic, many of them are parasitic, that fits in with the lifestyle, doesn't it? How do we put those two together? How can we put those two together and say that it's not unusual, or it wouldn't be out of the question to expect something that's anaerobic to be an internal intestinal parasite? What's the connection, do you think, between the environment where they're found in and the fact that they engage in anaerobic respiration? I mean, anaerobic respiration is not as efficient, right? You only get a couple of ATPs, but why would it be all right if you're a parasite? Or why would it be necessary, perhaps you're a parasite? One thing, yes, need less energy, that's good. Of course, you don't need to move around. Anything else? Yes, absolutely. There will probably be less oxygen down there, right? You might also absorb nutrients from the host, right? You might actually engage in diffusion of nutrients, of energy molecules across the cell membrane from the host, and therefore you get enough of it so you can engage in anaerobic and still survive. So yeah, nice work, folks. All right, so don't drink clean water, and certainly don't eat yellow snow. Now, the next one of excavata are the parabasalids. We looked at those in class the other day. This was the Trichomonas vaginalis. It was dyed purple, wasn't it? Yeah, it was. It was dyed purple. Remember I said they move two ways. They move with a flagella. There it is, at the front. And then these undulating membranes. And I said in class, it's kind of like the way a stingray swims. These organisms, of course, like I said, can cause vaginitis, a sexually transmitted disease. Silent males, remember, it's not symptomatic. Actually, one of the most prevalent parasites in North America, 5 million cases a year. It's not deadly, of course, and being the fact of its silent nature probably contributes to its prevalence. If you don't know you have it, you don't know you're spreading it. Once again, many times these are parasites or symbionts living in the gut, so they might have another degenerative mitochondria. This time we call it a hydrogenosome. Hydrogenosome. Holy cow. Hydrogenosome. There we go. Tripping over my tongue today. We find these kind of critters very interesting in the guts of termites. You remember back in Bio 1, you learned about the fact that like cows or animals, we can't digest cellulose. We can't digest it. We can't break that bond between the glucose monomers. But the bacteria that live in the gut of cows, they can. And it's the same thing with termites. Termites have parabasalids that live in their gut that helps them digest cellulose. So that's a symbiotic relationship. As a matter of fact, one of the byproducts of digestion of cellulose on the termites, and the cellulose, of course, is coming, if they're in your house, from the wood of your house, is after they're eating your wood, they become very, termites are very flatulent, like cows are very flatulent. So termites release a lot of methane from their digestion of cellulose. They release so much methane, by the way, that them and the cows are one of the largest contributors to methane in the Earth's atmosphere, which is one of the global warming gases, the warming, causing the warming effect that we see on Earth. Isn't that amazing? Must be a lot of termites out there. Must be a lot of farting termites and cows out running around. That's all. All right. Any questions about the parabasalids? So the excavata is the diplomonads, the parabasalids, and then the last group you probably know quite well, or at least relatively well, right? All right, the last group is the group we call the euglenozoans, and in that group there is the typical euglena, and then there's a couple of other not stranger, but different kinds of euglenozoans. But let's look at there's the typical euglena. Notice it's got the flagella. It actually has two flagella, one really small one, right? It's one really small flagella, and it also has one really short one, which you often don't see it. This is obviously an electron microscope picture that's been color enhanced. They are green. But there are a couple of cool things you can notice about it. One is the flagella. The other one is the wrapping that you see going around it. That's a band of protein that provides strength. Under the microscope, one of the ways we identify or help students identify live euglena is they're really twisty. They can move all around. They can fold in half. They're kind of like a ferret, right? Anybody ever had a ferret as a pet? Ferrets have that ability to bend. Yep, they bend, don't they, Sean? You can bend them quite a bit. They're very flexible. They can turn on themselves. It's because of the way their vertebrae are made. So that's kind of like the way euglena are. And that protein band that goes around them, yep, yeah, like a slinky. Like a furry slinky. Oh, my goodness. But that band around them provides this strength and flexibility to move around the way they do. Not at night. The one I had, they would get the kitchen cabinet doors open at night. They didn't latch very well, so it would bat at them until they'd open. Then would crawl on there through the pans at night and make all this racket noise. I thought for sure the first couple times when I had it that it was a rat. Open the door and here comes the ferret. All right, so these euglenozoans, many of them are mixotrophic. What's that mean when something's mixotrophic? Let me spell the word out. Mixotrophic. What's that mean when you're mixotrophic? Caitlin? Patrick? Anybody? Mixotrophic, right? We got autotrophic, which are like plants. Heterotrophic, you and me. And then if you're mixotrophic, what do you think you are? Look at the color of that euglena there. There you go. Euglena are green, so they're photosynthetic, right? They can photosynthesize and they're heterotrophs. What a great way to live. What a great backup to have if you don't have enough food. All you have to do is let the sun come up. They reproduce by mitosis, so genetic variability is not going to be inherited or passed down quite the same way that we're used to when we talked about it back in evolution. But remember, every time a cell divides doesn't mean that the DNA is exactly the same, and there's also this idea of viruses, and viruses and things can move genetic information between prokaryotes as well as eukaryotes. Within this group, there are some free-living forms, but there's some really interesting parasitic forms. So I'm going to talk about a parasite, one of these parasites that belong to this group in just a second. One interesting thing about these critters is something called a crystalline rod. You can see it in the picture down below. It forms a part of the structure of the flagella, but we're not sure exactly what it does. So there's a lot of mystery among the protozoan group. It has something to do with flagellal structure, but nobody's exactly sure quite the way it is, quite exactly what it does. Let's look at a parasitic euglenozoan first. All right, you looked at this guy under the microscope. This is the trypanosome down there in the lower left. Remember I said these are blood parasites. When you're trying to remember these organisms for later on, remember there were only 2 blood parasites that we looked at. There was this one, and then there's the one that causes malaria. This is not the one that causes malaria. Remember, malaria is carried by a mosquito. These kinds of diseases that insects very typically move between hosts, often they can be a bacterial. In this case, they're protozoan. Those organisms that move them around are known as vectors. So the mosquito that gives you malaria is the vector. The fly that you see in the picture here that is going to give you African sleeping sickness, the tsetse fly, is a vector. Blood parasites, the reason why they're good vectors is when you inject your blood parasite, your mosquito, flea, one of them, the big tsetse flies. When you're a blood parasite, what you do is you inject your proboscis, your pointed snout, into the skin of the host, and then you suck the blood out. But of course, what the blood is going to want to do is coagulate, right? That's what blood does. That's what capillaries, that's what blood vessels do when they're punctured. So what the blood parasite must do is it first secretes saliva or solution into the blood of the host that liquefies, that is an anticoagulant, okay? And then withdraws its blood meal, and when it's done, then it flies off. The itching that you feel from the mosquito many times is an allergic reaction to that stuff that they injected, the little bit that they injected to make your blood flow so they could get their meal. And so that's the basis of them spreading the disease. When they withdraw the blood from an infected host, say a host that's got blood and these trypanosomes swimming around in there, right? When it draws the blood from the infected host and goes to the new host, it injects a little bit of that back into the host to start the blood flowing, and there's where the parasite gets into the host, or it's into the new host, right? So when that tsetse fly goes from one organism that has that, no, it's the allergic reaction that you get. Yeah. So if that tsetse fly landed on somebody, took a blood meal, then went to somebody else, it would inject some of those trypanosomes into the other organism. And that's how it's passed. That's the African sleeping sickness. One of the things about African sleeping sickness it makes you very lethargic, hence the name, that makes it very difficult to control is the trypanosome is constantly changing the outside coat. Remember how your immune system recognizes things, right? Feel. Remember bacteria did this, didn't they, right? They made a capsule. Streptococcus did. The organism causes made a slime or a capsule over its outside cell membrane so that your immune system was like, I can't feel anything. So in other words, can't see anything, essentially, right? This little trypanosome that causes sleeping sickness can do that. The other one, that fly is perhaps a little creepy enough if you have something about flies, but let me show you the other one, Chagas disease. The parasite you looked at was Trypanosoma cruzi. It was the one that causes not sleeping sickness, but Chagas disease. Chagas disease is again spread by an insect, but it's not something, I was going to say not something as pretty as the fly. This ain't pretty at all. Take a look at this guy. Unless you're into bugs, then it is kind of cool. This is what we call the triatomy bug. These are kissing bugs, and what kissing bugs do is they basically will come down onto the host at night a minute. When you're sleeping, and they will, yes, and they will bite and get their blood meal, all right? And when they're done, as if it isn't bad enough that they've done that, this is a woman, you can see where she's been bitten by the kissing bugs. They around the mouth they bite too. That's the kissing part. But after they're done biting you, like I said, that wasn't bad enough. They turn around and defecate in the bite they just made, because in that defecant is going to be their eggs, and that defecant and those eggs are used as a starter pack so that those young can develop. Right? It's bad enough you bite me while I'm sleeping, and you give me this nasty bug crawling on my face, but then you crap in it when you're finished in order to reproduce. Thank you, Mr. Insects. Parasitic, right? Chemetoblastids. Any questions on those guys? Aren't those cool? Latin America mostly, Chagas disease. There have been some reports in the States. I was kind of looking at that online before class started, but it's not prevalent. It's one of them there tropical diseases, right? Yeah, kind of. You know, we use that host and the young. There's actually not too much. There's not too much science fiction in any part about that movie, as far as that goes, when it comes to organisms using spiders. No, it's wasps. Oh, which kind of wasps? Can't remember the name right now. But what they do is they'll take a spider, catch it, and they'll sting it, not kill it, but paralyze it, and then bring it back and wrap it up in a web, lay its eggs on it. It's still alive. Keep it paralyzed. And then the young of the wasp will, when they hatch, will feed upon the spider while it's still half alive. Well, it's being paralyzed, but it's still alive. It's important for the young to feed on living prey. Lovely, huh? Don't you wish you was an insect? Okay, the Euglena, as I said, two anterior unequal flagella that pull the organism through the water like an airplane propeller, essentially. There's the short flagella. You often don't see it because it's in that little pit. Being that they're mixotrophic, they're going to have an eye spot. So it's important for them not to, an eye spot, again, back to these things about, acceptations. It's not that they can see with it, but they can sense light. And if they're aquatic and they live in the water, that makes sense to have something like that. They have contractile vacuoles, okay? So what kind of environment do they live in? What kind of environment? Would they live in a hypertonic, hypotonic, or isotonic environment if they have a contractile vacuole? There you go. The word we use for doing that, for balancing your water, really cool word. I've got it up there by the arrows, called osmoregulation. So we could use that in a sentence by saying Euglena use contractile vacuoles to maintain osmoregulation. Other critters do different ways, but that's how you maintain that water balance. I already talked about the pellicle, and you should expect the fact that it's obviously got chloroplast. Now, what's interesting about that is it goes back to what I said about endosymbiosis. Because of the research we've done on these organisms, we think that the chloroplasts were originally free-living green algae at one time. They were engulfed by some eukaryotic ancestor of these organisms and turned into Euglena. All right, so a lot of that engulfing, like I said, was going on. All right, that's it for the excavata. Let's move on to the sarclade. Now, the name comes from, again, the fact that there's three groups within that, things called the stramenopiles, the alveolates, and then the rhizarians. So we'll go through each of those in order. We'll go through the stramenopiles first. Stramenopile actually means hairy flagellum. We look underneath the microscope and we can see why it looks like. They have a hairy flagellum, they have a smooth flagellum. In some cases, there are stramenopiles out there that have smooth flagella, even though the characteristic is to have hairy flagella. But the reason why is because they had hairy flagella, which they then lost. All right? You can see there's a picture down below of the hairy flagella that they have. This is going to add more drag to the organism in its aqueous environment, allowing it to swim more efficiently. There's the critter on the left-hand side. I always think, to me, that looks like one of those loofah sponges from in the shower, doesn't it? Doesn't that look like, or not one? No, not the loofah things, but my wife has those pink things that she cleans with, instead of a wash rag. Stromatophiles. Among the stromatophiles, there's a group we looked at under the microscope, the diatoms. Diatoms are fascinating. They comes in all kinds of shape. They're you saw just basically bits and pieces of them under a microscope. 100,000 species. Unicellular critters, photosynthetic, divide. They divide very interestingly. Let me see if I can get my pen to work. They're put together like a shoebox. Like this one on the right-hand side, this thing right here. You notice it's got two halves to it. We call those halves valves. Another word for a half, like a clam has two valves to it, or two halves. But anyway, they're put together like a shoebox. And when they reproduce, all they do is the shoebox splits in half. And which half got the bottom builds the top. And which half has the top builds a bottom. It's kind of a unique sort of binary fission there. A lot of these critters can live in really, really deep water too. I'm fascinated. I did a submersible dive when I was in college. I went into the Gulf of Mexico, and we took a submersible down around 1,500 feet to the bottom of where we were in the Gulf of Mexico to look for this eel we were looking at. The things I remember most about is the fact it was pitch black down there, and there were these beautiful, bright red, brick-colored shrimp. They were gorgeous, and they were like moving in slow motion. It was incredible. And we also saw a six-gilled shark down there. There was a shark that was just kind of swimming from out of the shadows into the light and then back into the dark. The styrofoam cups there are a reminder for me to tell you, if you don't ever if you've ever had a chance, go over to the oh my goodness, Harbor Branch over in Fort Pierce. Harbor Branch Oceanographic Institute has a little tour, and they have some submersibles there, and you can take a look at the submersibles. And one of the things on the tour they show you is where they've taken a normal size styrofoam cup, which is mostly air, right? It's mostly puffed up styrofoam. And they've taken it down in one of these subs and let it experience the pressure. And what it does is it takes that cup and it just kind of shrinks it almost perfectly in proportion by squeezing all the air out of it. So an awful lot of pressure in water. For every 33 feet, for every 10 meters or about 33 feet of water, you add another atmosphere of pressure that you're feeling. On land, about on sea level, we're experiencing, because of the column of air above us, about 15 pounds per square inch. All right, so if I go down in the ocean 30 feet or about 10 meters, I'm now at 32 pounds per square inch. You know, it's if you're a scuba diver or been scuba diving or anything like that, you understand that's the concern. The pressure there bounce up very quickly. In the deep ocean, some of these organisms can withstand pressures up to the point of 33 million pounds per square foot. Now, I'm not going to do the conversion into square inches, but you can figure out that's an awful lot of pressure. If you think about some of the pressures that exist on some of these organisms down, like from the Titanic, you know, the Titanic is 2 miles down. So if you want to figure how much pressure that is, it's 2 miles is, I don't know, 10,000 feet, about a little over, say 11,000. 11,000 divided by 30 is, oh my goodness, I'm not going to be able to do this. But I started it. Let's do it. 11,000 divided by 30 times 16. So down on the Titanic, the pressure is almost 3 tons per square inch. Amazing, right? And these little critters can survive. One of the ways they can survive is the valves, the outside shells, are actually made of something, got a little rigidity to it, kind of like the cell wall thing with plant cells, made of silicon dioxide, sand, basically. If you have an aquarium, especially saltwater aquariums many times, they'll use something called diatomaceous earth as a filtrant. What diatomaceous earth is they collect a bunch of these diatoms and they grind them up, and it's the shells. It's basically sand for the most part, but it's got more surface area because of the way the organism put it together than sand does, and it does a really good job of absorbing impurities in the water. Does anybody know what we say when something's a sink like that? I understand a sink is where you wash your hands, but what I'm saying there, diatoms are a major carbon sink. during blooms, during times when the conditions are right and the diatoms are just reproducing like crazy. What's that mean when we say they're a sink, carbon sink? Or could be any kind of sink, except not the kitchen sink. 3. Amazing. Yeah, 3 tons. Good gosh. Or a place it goes. Yeah, there you go, Sean. Yep. A place where it's stored. What we're basically saying there is a fancy way, I guess, of saying that diatoms engage in a lot of photosynthesis. Because during photosynthesis, right, remember back in Bio 1, you take carbon dioxide and you make glucose with it. Carbon dioxide provides the carbon. So a lot of the carbon dioxide in the atmosphere is taken up by land plants, but there's a huge amount of diatoms out there, and they're one of the major sinks of carbon in the ocean, one of the major carbon sinks, especially when they start to bloom. Now, that's a good thing because that introduces carbon into the ocean, and organisms feed upon other organisms and all that sort of stuff, and that carbon gets recycled. There's that really cool word or line in the song by Crosby, Stills and Nash that basically says we're all stardust, right? When stars explode, one of the things they make is carbon, and carbon radiates out to the universe, and then it gets recycled. Some of the carbon in you might have been carbon that was in, I don't know, Genghis Khan. It always gets recycled. Wasn't I read somewhere that Genghis Khan, who was rather prolific as a leader when it comes to reproducing, folks of Asian descent, 1 in 500 can directly trace themselves back to him, one of his direct ancestors. So old Genghis got around, I guess. Not like the Wilt Chamberlain. Wilt Chamberlain's book said somewhere like he'd had like 22,000 different sexual partners. I'm like, holy cow. He's got the time. All right, so we're staying in the sarclade. We're still in the stromatophiles and the hairy flagella guys. We've got the golden algae. All right, there's the flagella. This is Dinobryon. That's a golden algae. Biflagellated. Some are mixotrophic. Some unicellular, some colonial. There is that thing again. That thing about living unicellular but colonial. You're still independent. Yeah, it is a little, right? 22,000. I think we calculated up one time, and it was something like average 2.4 a day different. I'm like, holy cow. Anyway, you had to have caught something, I would think. So these critters, when we get in, we saw it already in the biofilms and the things that the prokaryotes do. That's almost kind of like cells getting together in colonies, and we see that trend continuing here in the protozoans, don't we? We see the fact that you're a unicellular individual, but you sometimes get together to live colonial, and that could depend on the environment. And a lot of these are very much adopt sort of the situation, or at least the strategy. Remember anthrax, the anthrax spores? And I talked about the fact that if the environment is bad, if it's not good, if it's deteriorating, one of the things you can do is die, of course. But the other thing you do is you can make a spore, hibernate, essentially, kind of, right? So you can form a very resistant spore, or in some cases we can call it a cyst, and Dinobryon can do that. So they can form these cysts when the environment deteriorates, and then these cysts can then germinate when the environment's much better. That's the basis behind the amoeba that goes up people's noses. That cyst lives down in the muck of the water at the bottom of water here in Florida. And when it's really warm out, those are conditions great for the cyst to germinate. But what really agitates it or gets the germination process really going is people in the water kicking up the mud, and all those cysts are getting in the water and they're mixing with the water and they're just whoa, germinating. Look, there's a nose, and they crawl up your nose. They crawl up your nose and they find your olfactory nerve, and they inch their way. It's really kind of creepy, but they inch their way along your olfactory nerve until they get to your brain. So I got some pictures of those. We'll take a look at that later. There's the golden algae. There's the brown algae, the big, huge kelp. Kelp is a cold water species, so unfortunately we're not going to get those big forests of kelp like you see in the picture. When I was stationed out in California in the Navy, I did a little bit of diving. They call them kelp forests, and there's a very good reason. These things can be 30, 40, hundreds of feet long sometimes. They're very interesting, though. Again, multicellular, but not tissues. So again, this idea of you're kind of, straddling the ladder again there, Aristotle, right? You got these brown algae, they're photosynthetic, all right? We call them seaweeds. We call them seaweeds often, but they're not plants, not if you think about a weed being a plant. But they are always multicellular, a multicellular protozoan, right? Not a plant though. The little nematocysts, that's cool right there. Those are floats. That's how these things float, because of course, since they're photosynthetic, they want to float up. Now the ones in the picture there look a little bit green. Well, they look quite a bit greener than the other ones, but they can vary a little bit in color, but they're all basically brown algae. Brown algae are really cool too. That picture I got down below is of the beluga. Brown algae forest beds are really good places for nurseries. A fish and other species will come in and reproduce in these brown algae forest bed or forests, because there's hiding. There's a lot of places for things to duck and hide if predators come. Well, the beluga know this, so what they do is they wait outside of these forests of kelp, and they wait for some little young fish to get a little bit too gutsy or too adventurous, or make a mistake and get a little bit too close to Mr. Beluga there, and they feed upon it. That's what's the cool thing about diving in these kelp beds, is just the fact that it's just like from top to bottom there are fish, man. It's amazing. Sharks will whip through there too sometimes. Mako sharks are very well known. I luckily, well, no, I would have loved to have seen something like that, but I would have it would have been obviously pretty unnerving, I guess. But folks had told me that you'll be in the kelp beds diving, and a Mako shark will just come whipping through as fast. Makos are like miniature, well, not miniature, smaller versions of white sharks. They'll come whipping through really fast. Their idea is a little different than the beluga. They're a little less patient on feeding. What they do is they tear into this place hoping to catch something off guard. Oh, shark! And then you're dead, right? Belugas just kind of wait until you kind of come out to them. But think about that as a diver, right? Here you are diving in these kelp beds, which are forests, like I said, and the shark was whipping right by you. That'd be so cool. All right, let me review something for you again, this idea behind what we use for terminology. You remember that these algae here look like plants, but that's really not the reason, that they look like that. So we call things like the roots, we call them holdfast, we call the stipe, and we call the blade. Now, what I want to know is the similarity between the way brown algae look and plants. Is that a homologous or analogous kind of feature? What would you think? Why is that, Sean? Why not homologous? Sure. And of course, everything is related, right? And I even told you that some plants, yeah, there you go. Convergent evolution and analogous structures basically say the same thing. So when something is analogous, it's that way because of convergent evolution. The wings on an insect, the wings on a bat, the wings on a bird all occurred that way through convergent evolution. There's an advantage to flying. They're all analogous structures, okay? Homologous structures can sometimes be similar. The leg on a dog, the leg on a cat. They can sometimes be very different too, right? They can be the leg on a cat, the flipper on a whale, like we talked about. All right, very good. Yep, those are analogous structures that came about through convergent evolution. And yes, Aaliyah has it exactly right. It has to do with ancestry. Yes, there are protozoans that are closely related to plants, but not this group. We haven't gotten to them yet. So this similarity is purely analogous, okay? All right. One of the biggest characteristics, or one of the characteristics that I need to introduce you to, because it's something that we're going to talk about as we go through plants especially, is this thing called alternation of generations. First of all, let me remind you how we reproduce, and animals typically reproduce, all right? Let's start with humans for a good example. If you remember that basically what we do is we are a diploid adult with 2 N chromosome cells, and we undergo the process of meiosis, and we produce single-celled haploids, right? Either sperm or egg, but they only have half the number of chromosomes. And then two parents, of course, are doing that, male and a female, producing haploid cells. And then do fertilization. Anything new about this to anybody? Here's Junior. Anything new about that to anybody? Right? Meiosis, fertilization. That's how, when we think about animals, there we go. All right? And when we're talking about the number of cells, up here, this is a multicellular adult. And these are single-celled sperm or egg. Yes, you may make more than one sperm. You may ovulate more than one egg, but each one is an individual cell unto itself. All right? And they come together and they form Junior. Everybody's familiar with that. Okay. Now I'm going to add something to that, or I'm going to add a variation. But before I show it to you, I want to talk for a second about this idea of surviving again. We've discussed that a lot through our discussions about evolution. If you remember, there are many ways, there are many strategies to surviving in the environment. They all involve basically reproduction. And no matter how you do it, cryptic coloration, having wings, evolving eyes, flying real fast, being a little taller with your neck than anybody else, it all comes down to numbers, right? Remember I told you sea turtles lay 100 eggs in a clutch, right? And out of 1,000 sea turtle eggs, one of those eggs does the organism that hatches from it. So there's a lot more that hatch, but only one of those lives long enough on typical to make it back to reproduce itself. And of course, if you think about it genetically, unless you reproduce, and I think I've said this before, your genetic information is gone. And the whole idea behind natural selection is to pass on genetic information, right? Advantageous genetic information. So numbers are what it's all about, right? Well, there are organisms out there that have taken this idea of numbers and applied it a little bit differently. So let me introduce you. This is what we call alternation of generations, and the organism we're looking at is the one we looked at in lab. This is the flat one, flat like the top of a counter. The leaf is very flat. Patrick, I know you and I talked about that. Do you remember the name of this organism? Remember I showed you in the jar, and I said the way I remember it is because it reminds me of a flat laminated top. The leaves do anyway. Anybody else want to help Patrick out? No, not the sargassum. Sargassum is curly, kinky. Remember the word sargassum, got a lot of S's and G's in it. This is the one in which, like I said, the leaf is like a flat laminated top. It might have been where it got its name to begin with too, right? Anybody remember what it was? Do that associative thing, right? That's what I'm trying to get at here. Look at the organism, flat. It's like a laminated top. This critter is called. Right? Laminaria. This is laminaria. Right? Laminaria. The one with the fingers, the one that was a brown algae that was in the jar, and there was one in one of those little plexiglass things. That's fucus fingers. Laminaria flat, fucus fingers. Got projections on there, okay? All right, so what you were looking at in the jar is one of these little leaves of laminaria, because that's what happens. These things grow and then they make more leaves. Alternation of generation. Let's start with what we know first. Let's start with this diploid thing right here. So this is the adult. Now, there are some terms here that we haven't seen before. In the alternation of generations form of reproduction, the diploid adult is known as the sporophyte. All right? Sporophytes make spores. Don't forget that. It's going to become very important because it's going to get confusing, because this occurs in plants and somewhat in fungi, but there's variations to it. But always remember, the sporophyte is diploid. The sporophyte makes spores. And since this is the adult, in the case of laminaria, unusual for protozoans, the sporophyte is multicellular. All right? So the sporophyte is diploid. The sporophyte is multicellular. The sporophyte makes spores. See the sporangia? The sporangia are going to make spores. Okay? Now, those spores are going to be created by meiosis. Now you learned about meiosis in Bio 1. I know you did, but I'm gonna add a level of your understanding here when you think about it in reproduction. Whenever organisms undergo meiosis, there is a reason why they're doing it. Variation. When cells divide by meiosis, the intent is not to make more cells necessarily. It's to make different cells. It's mitosis that makes copies of cells, right? So mitosis is about numbers. Meiosis is more about differences. And if I have to go into a discussion of differences and variability, then you haven't been listening to me since the beginning of the semester, right? So right there where I put that little blue T or whatever the heck that's supposed to be, what is happening is the sporophyte, cells in the sporophyte, in the sporangia, are making these haploid things called zoospores. What's a zoospore? Well, it's got a flagella. Things that can move, which are typically animals, will often have the word Z-O-O in it, zoo, like where you would go to see animals, right? So they get spores, no flagella. Zoospores, they have a flagella. These organisms live in water, so it makes sense to have a flagella. That's how you're going to get, that's how these zoospores are going to get out and do their thing. Now, here's where it gets a little bit wonky, so pay attention to what happens here. Here's where it's very much different. You would expect fertilization to occur next, but you notice fertilization a little bit further down the road. Before fertilization happens, those spores germinate, all right? And depending on the species, but in the case of Laminaria, some of the spores germinate and form these things called male gametophytes, and some make female gametophytes. All right, now here's where it's different. We make single-celled sperm that are haploid. We make single-celled eggs that are haploid. Gametophytes are haploid, but they're multicellular. They're multicellular haploid structures. Very, very big difference. Very big difference. The reason why this is called alternation of generations is because there are two multicellular stages. There's the sporophyte and the gametophyte. We, us animals, do not do that, do we? Right? We don't have a multicellular sperm. We don't make multicellular eggs. These organisms make these multicellular gametophytes. Now, there's a couple of reasons why they do that, and I'm gonna actually kind of go through these reasons more than once. So if you miss the significance of it the first time, when you're haploid, all right, you don't have to worry about genes being covered up. You don't have to worry about recessive alleles. There are no recessive alleles in a haploid cell, correct? You understand what I'm saying? Does that make sense? Haploid cells, the idea of dominant and recessive alleles doesn't happen. Because there's only one. So if you think about it, covering up genetic information, making something recessive, in some cases with some organisms, the way they evolutionary went about, that would be bad. Being diploid for these critters would actually be something that they would, I don't know if that's good. So when they make these multicellular gametophytes, one of the things they're trying to do is the fact that they're actually making extremely variable cells to deal with extremely variable environments. Now, if you're thinking, well, it must be diploid that's more successful, I remind you that a lot of organisms that have gametophytes, all the plants, by the way, are very successful. You got to keep in mind, just because we say, well, there are advantages to being diploid, doesn't mean that it's one versus the other. There are advantages to being diploid. There are advantages to being haploid. Thank you, Patrick. See what I'm saying? So don't say, never, ever think about evolution as getting towards perfection in any of these lines of organisms we're talking about. It's what works really well right now. And for these critters that do alternation of generations, because of the plants and these protozoan organisms here, this whole idea of making these gametophytes and leaving these cells haploid to have access to all their genetic information has worked for them. They've been around longer than us. All right? Now, another thing that gametophytes do, being multicellular means you can make lots and lots and lots, and I'll keep going and keep going. You understand what I'm saying? Sperm and egg, right? You can produce lots and lots of gametes. Why do you want to make so many? Why so many sperm, Mr. Gametophyte? Why so many eggs, misgametophyte. Why so many? Why so many? What happens to most of them? Yep. Right? Most of them aren't going to make it. Some do. And we're back to where we were. Yep. Most die off. Most never find each other. Remember, they're swimming around in water. We're a little different. We deposit the sperm kind of, you know, at the party. You know, these sperm and egg are mixing around in the water. Yeah. Yeah, exactly right. Exactly right, Sean. The more you have, some are going to die off. And you got to, and if you're having more than everybody else, that's what differential reproductivity is about. Okay? Fertilization, then sperm and egg come together. There's the zygote, and then the zygote grows through mitosis to make this sporophyte again, right? So everybody understand the differences between alternation of generations and, you know, say the way we produce. The big thing is that haploid stage, the part that's in the blue. We're going to have a multicellular haploid structure called a gametophyte. Gametophytes, alternation of generations, sporophytes. I bet you I say those words 1,000 times between now and the end of the semester, because we're going to talk, especially when we get into plants. One more thing, and I'll get off this slide, though. If the generations look alike, they're called isomorphic. And if they look different than each other, they're called heteromorphic. So if the gametophyte looks like the sporophyte, then it's isomorphic. And if they don't, then it's heteromorphic. Okay? Simple enough. Any questions on that? All right, so now we're going to move on. We're in the A part of the sarclade. So we've gone ourselves through the stromatophiles, the diatoms, and the two algae. So now we're going to go to the A's, and the A's are known as the alveolates. And the alveolates get their name because they have these little alveoli right here. Where in human beings would we find alveoli? That's pretty good for my right hand to color that in. There's a big one over here. Where in our bodies in humans would we find alveoli? Alveolus, I think, is the singular. Alveoli is plural, if I remember right. Maybe it's the other way around. Nobody took A and P in high school or anything? There? You have sacs in your lungs, air sacs. They're called alveoli. If you're someone who suffers from, oh my gosh, breathing problems, right? What's the name? What's the word I'm looking for? Asthma. Yes. If you're someone who suffers from asthma, what happens sometimes is your bronchial passages will seize up and tighten, and what that does is it prevents the air from getting down to these air sacs. Alveoli is where the gas exchange occurs. They get smaller and smaller and smaller. So it's a sac, okay? Now, alveolates and protozoans, don't use them for breathing, okay? It's a flattened vesicle is an alveoli. We give them that name, the alveolates, because everybody in this group, if you look at them, have these alveoli up near the front end, up near the flagella. Remember the flagella on the front on these guys. Now, for many years when I taught this class, we didn't exactly know what the alveoli did in this particular case. I mean, we know what they do in lungs, but we didn't know what they did with these guys. So, it's kind of like the whole idea of science changing, you know, those crystalline rods and euglena. Since then, we found out that these alveoli seem to function like Golgi bodies or Golgi apparatus. And I'll talk a little more about that in just a second when I introduce you to an alveolate and how they use their alveoli. But I want to remind you that Golgi bodies, remember, are involved in secreting material outside the cell. They can also disseminate material inside the cell, but they also are responsible for making those vesicles that deposit things outside the cell, right? Okay. Before I get to that one, let's talk about an alveolate that you guys have maybe come across and being here in Florida. Dinoflagellates. This is, change for me. There we go. Oops. Dinoflagellate. Dino means whirling. Dinoflagellates have two flagella, and so they move by spinning. That's where they get their name from. Most of them are photosynthetic. You can be red and photosynthesize. All right, you don't have to be green necessarily, depending on what pigment you're using. Unicellular, like a lot of other protozoans. Shells this time made of cellulose. So we saw the diatoms had shells of silicon dioxide. Dinoflagellates have shells of cellulose. There's some convergent evolution going on there, right? These shells, again, protect them. Dinoflagellates are very often found in aquatic or marine environments. They're what we call planktonic. And I need to stick on this word for just a second. We sometimes misunderstand what it means to be plankton. We talk about these little microscopic things that are plankton. Well, fish can be plankton. Essentially, you, dead, are plankton. What I mean, plankton are things that float. That's what they are. That's their definition. So it could be little dinoflagellates. It could be little plants. It could be a dead body. It could be a small juvenile fish. So that's what that means. The opposite of that, organisms that can swim around are known as nectonic. So fish, sea turtles, whales, humans when they're alive would be known as nectonic, all right? So dinoflagellates are one of those floaters. I want to make sure you understand that because plankton doesn't refer to a particular type of critter. It refers more to a lifestyle, right? A floating lifestyle. Some of these dinoflagellates can engage in that really cool bioluminescence. You've ever seen that? You know, where they get with the symbiotic bacteria, and they inside the dinoflagellate, when the bacteria get agitated, they'll light, they'll glow. Beautiful. I remember being in the Navy and seeing that at night. Like the ship would, especially when the water was calm, and the ship was cutting through the water and making that V wake. You could see just as far as you could see out to the horizon, this blue right on the wake where the dinoflagellates were getting. They weren't. The bacteria in the dinoflagellates were getting agitated, so they were lighting up. I'm going to stop on this slide, but I do want to mention this last thing. This is the Florida part. It is a dinoflagellate that one you see in a picture that's popular. It's known for the red tides that we sometimes suffer here in Florida. It's got a red color. And so during periods of blooms, when everything's wonderful and these guys start reproducing, so that could be, temperature. Happens a lot in the summertime here in Florida, so warm temperatures egg it on. Certain nutrients like fertilizers getting in the water will cause these guys to bloom, and they get so many they form this red color. That's the red tide. The problem with that is in these heavy concentrations like that, the dinoflagellates are always there, but in these heavy concentrations during blooms, they naturally produce a toxin. And in these heavy concentrations, like these red blooms, that toxin concentration can be deadly to fish. And if you've ever been on the beach, when there's been a red tide event, there's lots of dead fish up and down the beach, right? Some of that has to do with the fact that the oxygen is being removed out of the water by all these dinoflagellates that are respiring themselves. And if you've ever been on the beach during a red tide event, you might notice there's a lot of folks coughing too. When the waves break, the toxin becomes aerosol in the spray of the water, and you inhale some of the toxin, and it irritates mucous membranes. That's why it causes the irritation that you get. So a bloom is of course good for the algae, right? They oh yeah, everything's great, let's start reproducing like crazy, but it's not necessarily. And of course, the problem with the bloom, especially when you're something like a dinoflagellate, is you eventually kind of bloom yourself out, right? You start reproducing so crazy, you produce so much waste material in your own environment. It's like crapping in your own backyard, or you just use up all the resources in that environment, okay? So sometimes good, sometimes bad. But if you're trying to make the most of this situation as it is right now, go bloom. All right, I didn't get a chance to talk about the really cool alveoli. Well, they're all cool, but I'll talk about those when we get together on Monday. What do we got going on Monday? Let me see, next week. So that's the 9th. So we'll finish up this chapter, start on plants. We're doing exercise or lab 5, which is plants. Oh, so we start our plant, yeah. Yeah, it might. It also might have to do with the way the bottom is, Sean. A lot of times the water color has to do with the way the depth and the way what the bottom's made up of, sand or all that kind of stuff as well. Let me